Oded Rechavi will tell us what's beyond the genome. Oded, please.
Thanks a lot. I don't know if I'll tell you everything that's beyond the genome, just a little bit. But thanks a lot, Tzachi and Doron and Amit. I'm not a researcher who studies the origin of life, but I am interested in these questions, so it's a fascinating meeting to attend. And I didn't really know what I will present. I have two main interests that are relevant. One of them is the RNA world, and the other is non-ribosomal peptides. I don't have enough, unfortunately, about non-ribosomal peptides to say much here, but just for everyone here to know that I'm very interested in the topic. So if you're interested to talk, please tell me, and then we can talk about it later. Today I want to talk about the secrets of the RNA world in C. elegans, and whether it's perhaps related to something ancient, some RNA world that doesn't exist anymore. I'm more interested in the RNA world of today that's here, and to know whether it can work independently from the DNA world, whether RNA, at least in C. elegans, in these simple nematodes, can mediate inheritance that is independent of the DNA encoded instruction, that's passed independently of segregation of chromosomes, of the segregating chromosomes, and doesn't necessarily talk or communicate with the DNA genome instructions. A lot has been discovered in C. elegans nematodes about the regulatory RNAs, starting from the work of Craig Mello and Andrew Fire and Victor Ambrose and Gary Ruvkon. And so the small RNAs that were discovered there were later found to be conserved in other organisms. And also the first demonstration that double-strand RNA is a substrate for RNAi was shown in these nematodes, eventually leading to the Nobel Prize of Fire and Mello. What they've shown in their paper, aside from the fact that double-strand RNA is the trigger for RNAi, for RNA interference, is that these small RNAs work non-cell autonomously, that they can move between different tissues. The story, according to the legend, when Craig Mello, who was the first to successfully inject, to pioneer microinjections in C. elegans, when he looked over his shoulder, his student's shoulder, he saw that even the ones that accidentally inject double-strand RNA to the somatic tissues, instead of the germline, get F1 descendants that exhibit the phenotype, which suggested that indeed there's a transfer of RNA between different tissues. Later on, they also did it more systemically or intentionally, and showed that you can just feed worms on bacteria that produce double-strand RNA to get systemic silencing all over the worms, not only in the site of ingestion in the gut, but also in distant tissues, and moreover, that the silencing also transmits to the germline. And this is and is manifested in the progeny for multiple generations. And this is really amazing because there is this basic dogma of biology, also known as the second law of biology, that says that only the germline can transmit information across generations. The soma, what happens in the soma stays in the soma, and somatic responses cannot be inherited. But we see that in C. elegans, the soma, via RNAi, can communicate with the germline and the next generations, transmitting parental responses across generations. Now, in their studies, they only studied in these papers artificial responses to synthetic, exogenous, and double-strand RNA, or hairpin-derived double-strand RNA. They didn't examine whether natural endogenous small RNAs or small RNA-mediated responses transfer transgenerationally as well. When I started working in this field, I found that worms transmit antiviral resistance in the form of small RNAs, and that this inheritance proceeds for multiple generations, leading to culminating in the fact that worms are virtually resistant to all known viruses, just a handful of viruses that infect C. elegans very unsuccessfully that we know of. There are probably others that we didn't find yet. We also know now that changes in the environment that doesn't necessitate any addition of foreign nucleic acids can lead to transgenerational changes in the population of heritable small RNAs, affecting many different traits and the physiology of the worm. So if you starve the worms, it changes the pool of heritable small RNAs and probably also involves stomach-to-germline communication, resulting in descendants that are different epigenetically, in that they are more resistant to starvation and live longer and also have a different developmental range. There are different groups that have shown that changing the cultivation temperature of C. elegans leads to transgenerational changes in small RNAs that again affect different traits. And recently, again, a number of groups have shown that exposure to pathogenic bacteria lead to transgenerational changes in heritable small RNAs that affect pathogen avoidance in the descendants for multiple generations. So we see that different environmental conditions can sculpt the heritable small RNA pool and lead to a very wide range of phenotypes that persist for multiple generations. We know a little bit about the mechanism that shuttle small RNAs between different somatic tissues in the worms, and also from the soma to the germline. But most of what we know is about exogenous double-strand RNA. There are at least 8 different transporters identified to shuttle double-strand RNA-induced responses between different tissues. But we don't know much about the shuttling of endogenous small RNAs between different somatic tissues or from the soma to the germline. We've shown in this Posner et al. Paper that manipulating the production of endogenous small RNAs in neurons in C. elegans lead to transgenerational changes via a reshaping of the composition of small RNA pools in the germline, affecting the expression of many genes transgenerationally, and that this also affects the behavior of the worms in the next generations. So worms that cannot transmit small RNA responses from the nervous system to the germline have descendants that are defective in chemotaxis in high temperatures. And we identified some of the genes involved. For example, it involves a gene that's called HRD1, which stands for heritable RNA deficient 1, which is one of the argonauts in C. elegans that physically carries small RNAs in the germline. We also identified one target gene, SAGE2, which is absolutely required for the transgenerational response that the parents have over their progeny's behavior. But we still don't know which specific species of small RNAs are the endogenous species that actually move between cells. Something very important and relevant to this conference also is that for transgenerational responses in C. elegans to be inherited, to happen, the worms need to amplify their small RNAs. So you need to know this about small RNAs in C. elegans. There are different types of primary small RNAs. These could be short interfering RNAs that derive directly from the double-strand RNA template, an artificial one. There could be endogenous siRNAs or piRNAs or ribosomally derived small RNAs. There are different types. All of these primary small RNAs that also come in different lengths and nucleotide compositions do not lead directly to gene silencing. What they do is they recruit to the mRNA template RNA-dependent RNA polymerases that use the mRNA as template to synthesize much more abundant secondary small RNAs. And these are the ones that get inherited, and they are carried by specialized argonauts. The worms have 26 different argonauts, 19 of which are real genes and not pseudogenes. And different small RNAs are carried by different argonauts. 4 of the argonauts have been implicated in transgenerational inheritance, but that's also dynamic, and different environments can change which small RNA is loaded on which argonauts. But the heritable small RNAs are the amplified ones. There are genes like HRD1 that are dedicated specifically for inheritance of RNA, not for RNA per se in the same generation. So for example, here in the slide, you can see in the first row, this is a germline of a worm that expresses GFP. And you can see that in the upper row, if you expose the worms to RNAi, then the parents and the progeny silence the GFP in the germline. In the bottom row, these are RNAi mutated. This is one of the argonauts where RNAi just doesn't work. But if you see in the middle row, if you expose these worms to RNAi, it works in the parents, but it's not inherited to the progeny, because this protein is required only for transgenerational RNA inheritance, and there are other genes that affect RNA inheritance specifically, small RNA inheritance specifically. Interestingly, small RNA inheritance is affected also by just physical, just by chemistry or physical conditions, not only by regulated protein-protein interactions. So the sorting of the small RNAs to different argonauts and the choosing of particular mRNAs for transgenerational silencing occurs in these liquid-like germ granules that reside outside of the nucleus. What happens is that as an mRNA is expressed, it leaves the nuclear pore and enters one of these germ granules. And if this mRNA is chosen for silencing based on different reasons, then in the germ granules, most of the factors that are required for RNA inheritance reside. The RNA-dependent RNA polymerases, many of the argonauts, and the mRNA serves as a template for the synthesis of many small RNAs. And then these small RNAs move back into the nucleus and lead to the position of chromatin marks. They guide chromatin modifiers to the DNA of the target, leading to its condensation and recruitment to the nuclear pore that resides closest to the germ granules where the small RNAs were amplified. And then the two alleles are paired, and they reside just inside the outside, just near the nuclear pore that will lead to this particular granule where more small RNAs will be made. And just this granule will inflate later on so that small RNAs will be made and the gene will be silenced transgenerationally. And we've shown that manipulation of the germ granules by changes to the environment or by using mutants that are defective in the different germ granule components lead to transgenerational changes that last tens of generations and show very interesting non-Mendelian inheritance characteristics and parent of origin effects. So it matters whether you disrupt the germ granules in maternal or paternal lineage. And by the way, a different set of small RNAs are inherited in the sperm or oocyte. Typically, RNA inheritance, which is triggered by double-strand RNA, persists for approximately 3 to 5 generations. There are exceptions to this, but this is on average what happens. This was also named by Andrew Fire. This was dubbed the bottleneck to RNA inheritance, although there are many cases where it lasts longer. But typically, the response lasts for 3 to 5 generations. In the absence of a better theory, people just thought in the early days that what happens is that the RNAs that are made by the progeny are just diluting across generations until they are gone. But this cannot happen because every worm produces hundreds of babies. So after a few generations, the dilutant factor would be just enormous, would never work. And I also told you that the small RNAs lead to their amplification. So the worms can make a new pool of small RNAs in every generation. What happens instead is that there is a mechanism that terminates transgenerational inheritance after a few generations. We identified this negative feedback loop and genes that make up a transgenerational timer, this is how we called it, that time the duration of the heritable response. And we identify genes that are required specifically for controlling the duration of the heritable response that we called MOTEC genes. MOTEC stands for Modified Transgenerational Epigenetic Kinetics. And in MOTEC mutants, you get responses that last a lot longer than 3 to 5 generations. Some responses can even appear to be completely stable and last hundreds of generations. So this is just a few examples of MOTEC genes with relatively short, small effects, but there are other genes with bigger effects. We also know that the duration of response is not affected solely by the dilution of the small RNAs, because you can affect the duration of ancestral responses by starting new responses. So if you take a worm, for example, that expresses GFP in the germline, and you silence GFP with RNAi, then the effect would last for 3 to 5 generations. But we've shown that if in the F1s you challenge the worms with a second RNAi trigger, targeting a completely different gene, it would extend the duration of the ancestral response, of the ancestral silencing of GFP, because the second trigger will again activate the RNAi system, leading to the extension of the entire effect. Moreover, you can just consecutively, in every generation, trigger the worms with more and more, challenge it with more and more triggers to extend the duration of these ancestral responses. However, if you start an RNAi response that would typically last for 3 to 5 generations, for example, silencing GFP, and then in the next generation, you starve the worms or put them in high temperatures or put them in high salt, so you stress them, this will terminate the ancestral response right away. So instead of lasting for 3 to 5 generations, it will stop immediately. And this depends on stress signaling. So the worms needs to know that it's stressed to terminate the response, which means that the response is very plastic and it can be extended probably by manipulation of this amplification mechanism. And I think that the capacity of the worms to use mRNA to generate more and more smaller RNAs independently of the DNA sequence is very fascinating and still poorly understood. Very quickly, we also noted that there's variability in transgenerational silencing in isogenic worms. So although they're exactly the same, some worms that derive from a particular mother that experience RNAi will show a strong response, while others will show a weak response. This will change the duration. And we studied this by following huge population of worms consisting of tens of thousands of worms and looking at all the individual worms, looking for patterns that would explain this, the rules of this segregation. And we identified 3 simple rules that explain how RNA is segregated between the descendants in a population. These are rules that are very different from Mendel's rules. It's not explained by, segregation of chromosomes in meiosis. Instead, there are 3 basic principles that explain how the response would propagate. And we found that there's a mechanism that intentionally generates variability between genetically identical individuals, which is dependent on this major regulator of stress, HSF1. So just like there's a mechanism for creating intentional variability in DNA sequence, there's a mechanism for intentionally creating epigenetic variability, and perhaps as sort of a batch hedging mechanism, or we don't know exactly. How do these herbal small RNAs talk with the genome? What's the relationship with the DNA? So first of all, small RNAs, as part of their life cycle, they enter the nucleus, where they guide chromatin modifiers to the DNA, leading to the deposition of certain chromatin marks. What mostly has been studied is K9 trimethylation and K27 trimethylation. And this leads to transcriptional silencing, and perhaps also to condensation of the genome, which is something that we have preliminary results that suggest happens, but it's not published yet. And people didn't know what's actually being inherited. Is it the small RNAs or the chromatin changes that they direct? Because if you treat worms with RNAi and you sequence their descendants, you can find small RNAs there for multiple generations, but you also find that the same chromatin marks are present in the progeny. So what's being inherited? Is it the small RNAs or the self-templating chromatin marks that can perhaps affect their own deposition across cell divisions, although we know that large parts of the genome, of the epigenome is reprogrammed in the germ and in early embryo. We now think that what's actually being inherited are the small RNAs and not the chromatin marks. And we know this because we see very strong inheritance, even stronger inheritance in mutants that don't have these chromatin modifications at all. So even if a mutant that doesn't have K9 trimethylation at all can still transmit silencing responses. And this is true for most silencing responses, and except when it comes for silencing of newly evolved genes. There, these repressive chromatin marks have some role, which is still not entirely clear, at recognizing these foreign elements and directing their transgenerational silencing. And it's also important to say that transgenerational inheritance is a very fastly evolving trait. And you see different machineries in different, even closely related species, because this is a mechanism for silencing and getting rid of foreign elements, transposons and viruses. We also know from experiments that we did and also from experiments that other people did, that you can start an RNA response leading to the silencing of a particular gene, and then in the next generation cross out this gene, wait another generation and introduce a completely naive DNA locus, and it would still be silenced transgenerationally, which means that the silencing can persist even when the chromatin of the target, when the target is not there anymore. Perhaps based just on RNA that can lead to the production of small RNAs. Moreover, you can also silence a gene, cross out the target, silence another gene, so you will boost the system, as I told you, with a second trigger, and then induce the gene again, and you will get a stronger RNA response. So the whole system is amenable to manipulations, even when the target is not there. Nevertheless, there appear to be limitations to the capacity of small RNAs to self-amplify in the absence of de novo transcription from the genome. Andrew Fire, in his beautiful paper, showed that small RNAs that when you silence a gene by small RNAs, it leads to the amplification of new small RNAs, but that these new small RNAs are very limited in their capacity to serve as template on their own. You necessitate always new supply of mRNA that will serve as template. This is a beautiful paper, and he proved it in a number of different amazing, very elegant experiments. However, the limitation of the study that he only studied silencing of somatic genes, and it is possible that in the germline there are other mechanisms that allow small RNAs to self-template, and this is something that we're studying in the lab now. What we do know is that for transgenerational inheritance to occur, you need to have germline transcription of the target. And this makes sense because if the gene is not transcribed in the germline, there's nothing. There are no templates there to serve as no mRNAs there to serve as template for amplification, and the effect doesn't continue to the soma. But it's more complicated than that. We see that in most cases, except for some very interesting exceptions, silencing progresses transgenerationally only when it comes to silencing of germline expressed genes. Somatic genes get re-expressed. There are some expression, but this is a sort of a reverse Weismann barrier that prevents the germline from communicating back with the soma that we don't fully understand. And we are performing a mutagenesis screen now in the lab to try to define the composition of this reverse Weismann barrier. And the last thing, this is the last thing that I will discuss very briefly, and I think this is very relevant also perhaps to people who study the origin of the world or the emergence of the RNA world and its replacement by the DNA world, perhaps conceptually it's tied, is can small RNA inheritance affect the hardwired genome? Can it change the axial sequence of the target? Well, this is one of the main questions we're studying in the lab now. We think that it might happen, for example, because long-term silencing of a gene would affect DNA repair mechanisms such as transcription-coupled repair, and therefore silencing of a gene will lead to accumulation of more mutations. And also because condensed DNA, as I told you that these small RNAs appear to condense their targets, is less accessible to damage. And therefore, again, mutagenesis rate could be affected. But we also know, and this is something that we pre-printed very recently, currently under review, that small RNA inheritance can affect genome diversity indirectly. Small RNA inheritance is crucial for transgenerational, for the transgenerational viability of the germline. Worms that are defective in RNAi exhibit a mortal germline phenotype where they gradually become sterile. And this is specifically true for regulation of sperm functions. When you disrupt small RNA inheritance, the sperm doesn't work as well. And we found—
But then 2 minutes, please.
Yeah, great. I need one. And what we found in this paper is that if you grow the worms in high temperatures, it changes the pool of small RNAs that regulate the sperm. And when the sperm functions are compromised, it doesn't fertilize the oocyte. And when the worm senses that it's not fertilized, it secretes a pheromone that attracts males that would mate with the worm, supplying their own sperm. And when you mate, you dilute your genome in half, so you increase the genomic variability. So this is a way for stress to affect the variability in the genome, which is adaptive under stressful conditions. And we showed this in lab evolution experiments. So this is it. And thank you very much. And of course, I'm very, very happy to answer any questions.
Thank you very much, Oded.
Yeah, we have like 2 questions right now. The first is how RNA, how much are they stable in the cells, or are they degraded by ribonucleases?
Yeah, so they are degraded. First of all, we assume that if they're not protected by proteins that carry them, that they will be degraded right away. We don't know enough about their turnover in single cells or their levels. The stoichiometry of the entire thing is unknown. This is something that we're trying to address now, for example, by sequencing small RNAs from just the fertilized egg. But most of the studies have been done on RNAs from entire worms, so we don't have the numbers. And this is a very important thing or the turnover, the kinetics of the life of the small RNAs inside cells, and this is a major goal for the future.
Mm-hmm.
But there are also active mechanisms that degrade them.
Okay, great. And the second question I have here is, do you know of any example where RNA is being transferred laterally between worms, not specifically transgenerationally?
Yeah, so many people have looked at that anecdotally. So everyone who does RNA said, let's look at untreated worms and see whether RNA passes to these worms as well. No one has seen it, but no one published about it. So it should be done properly. Very recently, there's a preprint that has been published by Colin Murphy's lab, suggesting that worms secrete RNAs in a capsid of an endogenous retrotransposon that transmit a fear of bacteria, of pathogenic bacteria. The problem there, first of all, it's just a preprint. Second, the readout, it's a behavioral assay, so it's really hard to understand how it works. It's very amazing, but still controversial, and we want to see whether this what's actually being transmitted there and whether this is not some indirect effect or something. But there's one example which is very, very recent that I just described.
Mm-hmm.
So I'd like please to ask a question too. And since I see on the screen you, Oded, and Doron Lancet, I want to ask a question that relates to something I learned already at the times in Doron's lab 20 years ago. So you said curiously that for the gene to be silenced, it needs to be expressed in the sperm, in the germline, in the sperm.
In the germ. Not necessarily the sperm, but the germline.
In the germline, sorry, yes. And Doron probably remembers and knows very well, and other people might know too, that already 20 years ago, when olfactory receptors were discovered to be expressed in the epithelium of the nose, they were curiously found to be expressed also in sperms. That was interesting. Later, they were found to be expressed in many other tissues. And in addition, later it was found that many other genes, not olfactory receptors, are expressed in the sperm. So the question is whether we can be bold enough to connect between the dots and suggest that perhaps, and I think, by the way, Tai and I has a recent Cell paper that
right.
shows massive expression of genes in the sperm, suggesting that this could somehow facilitate some mode of evolution. But maybe we can be brave or not to speculate that this could be related to what you're saying.
Yeah, I think it's a great question. And yes, Itai and I indeed says that the sperm expression is sort of a playground where you test the expression of different genes and also make them more or less amenable to acquiring mutations. Right. I think it's very, very interesting. And it's specifically interesting with the odoreceptors because, I mean, the worms, most of what the worm know how to do is to find food using its many, and I think it dedicates the largest portion of its genome, any other organism, for olfactory receptors, 1,300 olfactory receptors. Right. Normally, when I think about the type of responses that can transmit transgenerationally, I think about, very general responses, sort of like an inflammation or a general capacity to be stressed or something like that. But in theory, by regulating an olfactory receptor, you can control the response to a very particular odor or an element in the world. So it's possible that you express certain genes to allow their silencing transgenerationally. I think it's a very bold hypothesis and a very interesting one.